All right. Now I'm going to pause here, see if you have any questions, and then we're going to move into actually looking at the process, the steps, if you will, of glucose oxidation. So we've talked about it from a big picture standpoint. We've looked at the equation. We've talked about the fact that a lot of that energy is not actually captured. Here, we're going to move on to look at what's exactly happening from that single molecule of glucose to result in our 38 ATP. Okay. So if we look at glucose oxidation holistically, the first two steps, which are glycolysis and the Krebs cycle, they're going to make a small amount of ATP, and they're going to do that by substrate level phosphorylation. Now, going back to my initial analogy, this was akin to borrowing phosphate or taking a phosphate from another molecule in the cytosol. Okay, and in the cytosol is where we have glycolysis and the Krebs cycle taking place. Now, it's not going to release a lot of ATP. It's going to be a very minimal amount, but it's not going to require oxygen. So it's going to be somewhat of a beneficial scenario to be able to make a small amount of ATP very quickly without having to input any oxygen. Now, the third stage here, this is oxidative phosphorylation. This occurs by the electron transport chain. And again, this is what makes the bulk of our ATP. So of the 38 ATP that we liberate from a single molecule of glucose, about 34 of those ATP are going to come from this third stage, which happens in the mitochondria and is going to make ATP. But again, it's going to need oxygen. The whole reason that we breathe in oxygen is to drive this process. And so this cannot happen in the absence of oxygen, which we can think about as somewhat of a price to pay to make a whole lot more ATP. All right, much more of a sustainable source of ATP. Okay, so the first stage, glycolysis, this happens in the cytosol, very important to kind of set the scene of where this is taking place. And this is basically a series of enzymatic reactions to go from glucose to pyruvate. Okay, pyruvate is going to be somewhat of our product that is released at the end of glycolysis. So it's a sequence of 10 enzyme catalyzed reactions. And I don't need you guys to memorize the steps or the enzymes or the substrates in each of those steps. But what I do need you to memorize here is that we have one molecule of glucose to start, and we end up with 2 pyruvate at the end of that. What happens in the mix? What happens in the middle of this? We generate 2 net ATP. And we say net here because we had to input 2 ATP. So the cost of glycolysis is 2 ATP, and we end up generating 4 ATP. So we really only make a net of 2 ATP. What else do we generate? We generate 2 NADH plus electrons. Okay, so we have to input NAD plus, all right, which is in the oxidized form. Remember, oxidation is loss, reduction is gain. So if we have the oxidized form of these 2 NAD, and then on the other end of glycolysis, we're going to end up having the reduced form, which is where we've added 2 electrons to those NAD. And then we have no oxygen consumed, no carbon dioxide produced. Really important to make that note here, because we do need oxygen in that third step that we mentioned, and we will produce carbon dioxide in those later steps as well. So at the bottom of the screen, we can look at the overall reaction for glycolysis. We input 1 molecule of glucose. We have to also input 2 NAD plus in the oxidized form. We also need to have 2 ADP standing by. And then we've got 2 phosphates. Again, we're taking these phosphates from other molecules in the cytosol, hence the term substrate level phosphorylation. And on the right side of our equation, we have 2 pyruvate at the end per molecule of glucose, 2 NADH plus 2 electrons, and then we've got 2 ATP net. Okay, now also important to know here is that the electrons that we are generating here And then the other intermediate steps that I'll mention are going to the mitochondria to help with oxidative phosphorylation. So the electrons that we're making here have a really important role to play in that third phase of glucose oxidation, which is the oxidative phosphorylation via the electron transport chain. Okay. So here are the steps of glycolysis. Again, I don't need you to remember these or memorize these. If you happen to go on to grad school or med school, you definitely would have to know these step by step, substrate and enzyme of each step. But for the purpose and the scope of this course, I simply want you to know what we put in, 1 molecule of glucose, and what we get out at the end, 2-pyruvate, 2 NADH plus, and then 2 net ATP. Now, I want to use this slide to explain how we get the net in that net ATP. So the energy investment phase of glycolysis is the initial 3 reactions here. And we had to put 2 ATP in to power the enzymes here. So hexokinase, phosphofructokinase, those 2 enzymes need energy invested. So we had to put 2 ATP into the process in this phase. In the latter phase, the latter half of glycolysis, especially starting from G3P, glyceraldehyde 3-phosphate, here we start generating 2 of each products for the 1 G3P that we invest, right? And so the splitting of the substrates at this phase means that at the end of this, we're now going to end up having 2-pyruvate, although we only put 1 glucose in to start. We're also going to yield 2 ATP. So phosphoglycerate kinase yields 2 ATP and pyruvate kinase yields 2 ATP. So in our energy harvesting phase of glycolysis, we generate 4 ATP, but we're going to subtract the 2 that we had to invest, and that's how we get that 2 net ATP. Okay. Right. And then here's another much more fancy drawing. Again, peruse this, review this from more of a holistic standpoint, and not necessarily memorizing the fine details here. All right. The second phase that happens occurs in the mitochondrial matrix, and this is basically a linking step. So this is a transitional step that goes from glycolysis, our first phase of glucose oxidation, to the Krebs cycle, which is our second phase. Some literature refers to this as the third phase. For us, we're going to call it the second phase of glucose oxidation. So glycolysis, the linking or intermediary step, and then the Krebs cycle. So what exactly happens in this linking step? We've got our pyruvate that we generated from glycolysis, okay? And for each of those pyruvates, we are going to liberate 1 acetyl-CoA. So we input pyruvate here, all right? And then we're also going to input NAD plus in the reduced form, or the oxidized form, excuse me. And then we're going to have to input CoA, coenzyme A, and then we're going to generate at the end acetyl-CoA. Okay, acetyl-CoA. And acetyl-CoA is going to be our main product of the linking step, and that is going to go on to be the main substrate that powers the Krebs cycle, which is our second phase of glucose oxidation. The other things that we generate as a byproduct here are carbon dioxide. So notice we did not generate any carbon dioxide in glycolysis. In the linking step, we do begin to generate carbon dioxide. So our overall reaction at the bottom here is 1 pyruvate, 1 CoA inserted, 1 NAD plus inserted, and then on the other end of our equation, we have acetyl-CoA at the end, heading towards the Krebs cycle, excuse me. We generate some carbon dioxide as a byproduct, and then we generate some more electrons in their reduced form. Again, these electrons are going to be heading to the mitochondria towards the electron transport chain to participate in oxidative phosphorylation. So as we're speaking about each of these steps going towards the electron transport chain, the electrons that we're making in all of these preliminary steps are actually going to be really crucial to what happens in the mitochondria as a part of oxidative phosphorylation. Okay. Now, the other thing I want to point out here is that from glycolysis, we made 2-pyruvate. Here, we're only looking at what happens to a single pyruvate. So in the grand scheme of things, for 1 molecule of glucose, we generated 2-pyruvate. Therefore, we're going to generate 2-acetyl-CoA. This step is going to happen twice for every molecule of glucose. All right. I hope that makes sense there. Let's move on to the Krebs cycle. And the Krebs cycle is a cyclical step or a cyclical series of enzymatic reactions. And we say that because the initial substrate ends up being the main product at the end. So we don't have a different product from the substrate as we saw with glycolysis and as we saw with that linking step. Here, acetyl-CoA from the linking step is the main substrate for the Krebs cycle. And then that ends up being the main product as well because this is cyclical. The Krebs cycle, just FYI, is also known as the TCA cycle, the citric acid cycle. Those are all synonymous terms for the exact same process. This occurs in the mitochondria. This is going to produce ATP. Okay, it's going to also produce some carbon dioxide as a byproduct, 2 carbon dioxide per acetyl-CoA. And then we've got 2 NADH plus electrons. And we have another electron carrier appearing here for the first time. So this is FADH carrying electrons, which is in the oxidized form, excuse me, the reduced form. I have to keep reminding myself. So the reduced form, which is the carrying electrons. And then that was the product generated, and we had to insert 1 FAD, which is the oxidized form, not carrying any electrons, all right? So at the end of this, we have 1 acetyl-CoA inserted. We have to insert 3 NAD+, right? We have to insert 1 FAD, and then we have 3 molecules of water, 1 ADP and 1 phosphate. And on the other end of our equation, we liberate 3 NADH carrying electrons, 1 FADH2 also carrying electrons. Both of those are going towards oxidative phosphorylation once again. As a byproduct, we liberate 2 carbon dioxides and we generate 1 ATP. Okay. So again, we want to start putting this into perspective because we had 1 molecule of glucose generating 2 pyruvate per molecule. That's going to generate 2 acetyl-CoA per molecule. At this stage of the reaction, we now have 4 net ATP. Okay, and I want to pause here to make sure that we understand how we've gotten those 4 net ATP. 2 net from glycolysis, right? 1 ATP from each of our acetyl-CoA, right? Remember, we've got 2 acetyl-CoA at this point because we had 2 pyruvates per molecule of glucose. And so the Krebs cycle and the linking step had to happen twice per molecule of glucose. So we have 2 ATP from the Krebs cycle and 2 net ATP from glycolysis. So at the end of this, all of the ATP that we make in the absence of oxygen is 4 net ATP out of our 38 total per molecule of glucose. So a very minimal amount is made outside of the electron transport chain, outside of oxidative phosphorylation. All right. And again, here is some more details of the Krebs cycle. Again, I don't need you to memorize this. I just need you to remember where the step happens in the mitochondria, what's inserted, acetyl-CoA, what's produced, acetyl-CoA, and then everything that we make as far as the byproduct. 3 NADH carrying electrons, 1 FADH carrying electrons, 2 carbon dioxides as a byproduct, and then 1 ATP per acetyl-CoA. All right. The third step or the third phase is the most important phase, okay? And this is where we'll spend a lot of the rest of our time today and on Wednesday talking about the oxidative phosphorylation. Here is where we are going to make 34 of the 38 of our total ATP. So the glycolysis stage, the linking step, and the Krebs cycle only yields us 4 ATP molecules per molecule of glucose. The bulk of that, the rest of that, at 34, is going to come from the oxidative phosphorylation. How are we going to make that? We're going to take all those electrons that we've been sequestering, right? We've been gathering electrons in glycolysis. We've gathered some electrons in the linking step. We've gathered electrons in the Krebs cycle. All of those are being shuttled off into the inner mitochondrial matrix. And we're going to transport those electrons down a chain, literally the electron transport chain. And as we pass these electrons along this chain, we're going to take that energy. Again, remember, energy is never created, it's always transferred, right? So the passing of these electrons is going to yield the energy that is going to synthesize the bulk of the ATP that we liberate or that we capture from the food that we eat, right? From that single molecule of glucose. The caveat here is that we need oxygen, okay? Unlike what happened in glycolysis and what happened in the Krebs cycle, we need oxygen to make the rest of the ATP. And this is the sole purpose of us breathing in oxygen, is to drive the electron transport chain. Okay. So there are a series of molecules that are inserted in the inner mitochondrial membrane. And the molecules are going to undergo oxidation reduction reactions going from high to low energy. Again, oxidation being a loss of electrons. And if we reduce those electrons, we're going to, if we reduce those carriers, we're going to liberate those electrons. And so we can take that going from high to low energy, and we can harness that energy to make the ATP. And oxygen is going to be standing by to be the final electron acceptor and help drive that chain so that we can continue to make that ATP. All right. I don't want to get into this because I think it's going to need a little bit more time. So I'm going to pause here, see what questions that we have, and we'll pick up with this probably when we meet on Wednesday. Again, on Wednesday, we're going to skip over objective 2. And just to give you another sort of summary about what objective 2 is, it's basically how we can do exactly what we talked about today with glucose, but we can do that with other substrate as well. So we can break down fats, we can break down proteins. Proteins, I will continue to reiterate, is a very rare phenomenon that our bodies break down proteins. We have to be in starvation mode to really start to tap into proteins as an energy source. We typically usually break down carbohydrates as our main source, our primary source of making ATP, and then by other processes such as ketosis and fatty acid oxidation, beta oxidation, we can break down fats to make energy as well. So we're going to mainly focus on carbohydrates, glucose, which is what we're doing today. But objective 2 is going to go into how we can use other substrates as well. And again, I don't typically test this on the exam. It's more of some nice to know that we are using other substrates. What we mainly focus on is glucose, and that's what will primarily be on your exam questions from this unit. But have a read of the other content. The missing text is going to be in the slides for those portions. And then when we come to class on Wednesday, we're going to put this knowledge to the test. We're going to apply this with a case study, look at some practice questions, and then we're actually going to illustrate what happens with the electron transport chain and talk about exactly how we make this massive amount of ATP in the mitochondria with the presence of oxygen.